Okay.
Okay.
Hello. Good morning or afternoon, wherever you are. Thanks a lot for organizing this super interesting session. And I'm happy to talk about our work here, which is about a method where we combine array tomography with electron tomography. And actually, this project started when we had basically a CLAM approach to study the blood-brain barrier. And I'll basically walk you through the first steps we did and where we saw the necessity to then develop a method where we combined both techniques. So we are situated in Munich, Germany, and we are working with different groups in the field of neurobiology. So the hub is called the Nanoscale Hub. And yeah, here is some images of our facilities. And our interests are in neurodegeneration, neuroinflammation, and gliovascular dysfunction. And in the project that I will introduce to you now, we were interested in gliovascular dysfunction, namely in the the leakiness of the blood-brain barrier. And this can happen during stroke or, for example, also traumatic brain injury, which serves as a model system in our hands. So just briefly to introduce the blood-brain barrier. So there are several layers, cellular layers that ensheath basically the lumen of a blood vessel. So one is the endothelial cell layer, then there are pericytes, and then you also have the astrocyte end feeds. And of course, you have also luminal cells, for example, the erythrocytes. And during disease, like, as I said, for example, traumatic brain injury, you have destruction of certain cell types and migration of immune cells into that region. And this is all then mediated via this blood-brain barrier that has some leakiness. And the features of the leakiness are, most of these features can be observed by electron microscopy, like, for example, the tight junctions, of course, also with molecular markers, but electron microscopy is a very nice way to monitor leakiness of the blood-brain barrier. As I already mentioned, in our model, so we have a mouse model of traumatic brain injury with controlled cortical impact. And there we usually observe vascular occlusions at vessels that are partially damaged. This is shown here where we have certain cells, luminal cells like erythrocytes, but also platelets that accumulate here. And what we use to monitor that is fluorescent lipid nanodroplets, LNDs in short. And with confocal imaging, we then see accumulations of these nanodroplets at a certain site. And this is how the experiment looks like. So we do, basically our collaborators do the controlled cortical impact, and then the lipid nanodroplets are injected. And at the end, we also inject a lectin to just label basically the healthy blood vessels, so the whole vasculature. And this is how this experiment looks like. And here we were comparing lipid nanodroplets of different sizes, so 30 and 80 nanometers. And we basically found that so these sites can indicate, so these droplets can both indicate sites of vascular occlusion or of leakiness. But while the density was very similar, we saw less lipid nanodroplets extravasating these occlusions. So that means at the end, we came to this model that the 30 nanometer lipid nanodroplets, they monitor early events of blood-brain barrier leakage, while for the 80 nanometer droplets to really pass the barrier, the vessel has to be more destroyed or more heavily destroyed compared to the 30 nanometer ones. So we went on with these 30 nanometer particles as early markers of blood-brain barrier leakage. And also in combination with 18 nanometer particles. And the beauty of that is now that these particles can be visualized also in EM, and you see that there is some distribution of the particle size. of both 30 and 18 nanometer ones. So what we do here is to have a vibratome section and identify the site of vascular occlusion by confocal imaging, and then we do array tomography on this the site that we identified with light microscopy. And the technique that we apply here is automated tape collecting ultramicrotomy, where we have a tape collector attached to the ultramicrotome, and then the sections are collected onto the tape and imaged sequentially with a scanning electron microscope, and then we reconstruct the volume that was imaged by the SEM imaging. And then we can correlate the region that we previously identified by confocal imaging. So this is now the low resolution image, and you can see here that this is a occlusion with a very bright signal from this lipid nanodroplets. And with electron microscopy, we can now reconstruct the entirety of this occlusion site with all the fibrin that is clotting here the vessel, and we can also identify accumulations of lipid nanodroplets that are in the vessel or outside the vessel. However, what we found, or what you could also see in the previous image, that these lipid nanodroplets are not very so the size is not very, it's not unique, and the gray value is also different. So yeah, varying. So that's why we co-injected gold nanoparticles of the same size. So 30 nanometer gold particles along with the 30 nanometer lipid nanodroplets. So and this helped us now to really clearly identify the location of these gold particles. And this is particularly helpful if you want to really monitor single particles that are not in the lumen of the vessel, but outside the lumen. And yeah, with that, we can then reconstruct the entirety of this microclot. So where we see erythrocytes in red, platelets in blue, and then here you can see even a cell that is extravasating. So there's just undergoing extravasation here. And you can also see that we can now also monitor the single gold particles that are abluminally located. But what is still missing is, and what's the interesting part is basically the status before extravasation. So where do these particles really end up? And there is a limitation of the atom method, because we have like physically a section at 50 nanometer thickness. So that means we only see the particles at one section, but not on the next section. So we don't have a 3D information on the particles themselves, on the location of the particles. And then we thought it would make sense to separate this atom method into two parts, the screening part and the high resolution acquisition part. And then if we think we can also section thicker and image not with SEM, but with TEM tomography, we could use this screening phase to really locate here our region of interest. In this case, it's this microclot or this clotted vessel, and then zoom into the region of interest with high resolution. And then we can have high resolution isotropic voxels of the region of interest. So that was the plan. And what we tried first is to get the section of the tape, of the plastic tape, onto a grid, because that's required for the TEM tomography, but there was no way to get that off with any chemicals or any physical ways. And that's why we are using now coated tape. So that's the same tape, carbon coated Kapton, but it's now coated with a marker pen, and then we can cut off basically the gluing tape around the plastic tape, and then rinse the section off with acetone and collect it on a TEM grid. And this is how that looks like. So this is now the real section image in SEM, and then you can see two sections, so from a series of sections, and where we cut out one and rinsed it off the Kapton tape and collected it on the grid. And what we use for this is a standard marker pen, and we have this kind of a bit too fancy instrumentation for this, but you can just use your pen and coat your tape. So it's really super easy technique. And we plasma coat it several times after the coating. And you see why this is needed now here in this figure. So this is normal carbon coated and plasma treated Kapton tape. But it's not basically this our reference. It's not removed, so it's not removable from the tape. And then this is now with formbar coating. So this is something we tried before the marker pen, but you see that there is some charge. There are some charging problems, and we also had problems with that it was not really homogeneous coating. So the other thing that we tried is red. So a marker pen with red color, and there we also had huge problems with charging. And also if we have a black marker without plasma treatment, but black marker with plasma treatment works very well in our hands. And this is just a comparison of the ultrastructure of normal carbon-coated Kapton compared to our coated tape. So it's really nice ultrastructure. And here we show that you can now really have an overview imaging of this cortical region here. Then you can zoom into one region. In this case, it's a blood vessel. This is with SEM imaging, and then the same region is found in the TEM after removal of the section. So this is now an ultra thin section. And that also works with different resins. We tried Jerkapen and EPON. And so this is now the semi-thin section where we did real serial array tomography, serial sectioning, imaging, and segmentation. And this is a single SEM image, and this blood vessel found then in electron tomography with all the details, and also reconstructed then these details of that image. And we can also do that on serial sections. So George, who is basically pioneering this approach, he was able to collect here 8 subsequent sections, and you can see that here this is a very rough reconstruction due to the thickness of the serial sections, and this is the reconstruction of the serial tomography images. And yeah, you can see here that now we can see all our lipid nanodroplets in magenta and gold particles. And what we were interested in, that they are located in this endothelial layer. And we found that they are, for example, mainly located in some blabbing structures of this endothelium. There are some limitations of the method. So one is that the marker pen contaminates the section. We see that really at the surface of the section. So at one side of the section, and in this case, it affects the reconstruction, but it's really not penetrating into the section. It's really on the surface. And there's also limits to the imaging conditions. So if you go really high resolution with the SEM imaging and with high dwell times, then you cannot remove the section anymore because it's kind of stuck onto the tape. And with this method, we think that it nicely complements correlative techniques from basically clamp techniques, combining light microscopy and SEM imaging. So similarly, Atom Tomo can now combine SEM and TEM in a nice way and in an easy way. And finally, I also want to announce that we are part of an array tomography training and network initiative, and will host one of the workshops within that initiative. So the application is already closed, but if you're interested, we will have another workshop in Argentina in 2025 where we present this method, but together with a lot of experts in the field, also other methods and with hands-on practical. So stay tuned for the next workshop. And I'd really like to thank the people that were involved in this project. So mostly George, he did all of the array tomography work. Then Thomas Miskeld, who supported us also with array tomography. The electron tomography was done in Thomas Müller-Reichert's lab in Dresden, mainly by, or only by Gunnar Fabig. And confocal imaging and the mouse model was done by Igor from Nicholas Plasnilla's lab. And with this, I'd like to end the presentation and I'm open for questions.
Thank you, Martina. It's fascinating work that you did there. Is there any questions? Otherwise, I will ask something. The lipid nano droplet, what was the composition? Because it's not electron dense, but they do have some density in the TEM. So
yeah, that's from the lipid shell. That's what you see, and in the electron microscopy images, you can also see that it's very bright. So it's very hard to identify them. It's mainly if there are really accumulations of these lipid nanodroplets that you can really easily identify them because otherwise it's just a very faint circular shaped object. Yeah.
So are they very stable? These nanodroplet, the lipid nanodroplet? They don't degrade or anything?
Fixation? Yeah.
So we did fix that,
Yeah. So we so what is nice about them is that they are very bright, and also the lectin is bright, and it also survives basically the glutaraldehyde fixation. So it's you can fix it like for optimal way optimally for electron microscopy.
Okay, Melania, you have a question? You can unmute yourself.
Yeah, sorry, I don't have video today, but so I have a question about the tape treatments you're doing, the pens and the plasma. Did I understand you? There's some kind of plasma pen, or did I mishear that?
Yeah, no, I think that was maybe too quick.
Okay.
So we have the Kapton tape. That's the material. That's basically the same that you or I at least still use for cassettes.
Right.
So it also is this brownish tape, and then it's carbon coated. So that's the basic tape that we use for normal atom. And then for this, in order to be able to remove the section, we coat it with a pen. It's so the brand is Edding. So it's a black pen, and you basically only mark the pen like you would mark a piece of paper, and then this marked tape. We put this marked tape into a plasma coater.
Yeah, okay. That makes sense. Yeah, we have the system. This is really, I think it's really clever, and I wanted to know what that pen was, so maybe if we ever have time, we can try it.
Yeah, so as I said, it's not working with other colors because so we think that there is some kind of carbon in the black pen, and that helps with to avoid charging. But we don't. So we contacted Edding, but we haven't found out the exact composition of that.
Right. Okay. All right. Thanks.